Okay, hello everybody, and welcome to today's Biodiversity seminar. Today's speaker will be Pollock from McGill University. Laura leads the quantitative biodiversity lab there and is an assistant professor. She did her PhD in Melbourne, finished in 2012, and then did a small postdoc there, and then went on for a postdoc to Biodiversity's lab in Grenoble, and then 3 years ago started a professorship in Canada and works mainly on biodiversity conservation, species distribution, and climate change. That's the goal here today. So, Laura, first.
Can the people online? They should have come in person. So thanks everyone for coming today. And I apologize in advance because most people most people don't talk about today how to understand biodiversity. So this is the problem. Almost all species and why. So one of the questions that we're trying to address is how, so if you just do a standard species distribution model and make projections, etc. So if you run a standard species distribution model, put in whatever in terms of the number of species, and that's if everything tracks the climate. So clearly, this is not. Okay, is that better? After all that with my hair. Okay. So the big question is, we know these projections say everything's moving north. We know realistically, who knows how many of the species will move north, but not all of them, right? So a lot of the things we've been trying to do is understand how many species, what type of species are actually going to be moving, and therefore how vulnerable are these northern ecosystems going to be? But the other big problem in Canada is this. So this is a map of, can you see that even? This is a map of GBIF observations. So it's a 1 kilometer grid cell around the world. You can see Europe over there, completely lit up because we know every single, where every single thing is, right? Then you can see kind of the border of Canada, and especially the border of the boreal forest. So once you get into the boreal forest, we have almost no GBIF observations. And this is GBIF, even for birds. In almost all of that area, there's zero. There's no information, even for the most common species, but yet we know a whole bunch about the sort of the border area. So the other problem is if we run these models, we have so much uncertainty that in a lot of cases, it's difficult to tell what will happen. This is just another way to visualize it. So this is the climate velocity. So anything that's colored here at all are cells that have the highest 25% of climate velocity. So they're changing the most rapidly. And the color denotes how much of sample effort there is in each cell. Again, there's a lot of sample effort in the prairies, which is good because there's a lot of threatened species there. But for almost all of the area where we expect climate to be changing the most, we have zero GBIF observations. There are just a few, you can see a few of the northern research stations, and mostly what they do is monitor a couple of bird species. So this is what happens if you model the distribution. I was going to figure out what species this is. I still don't know, I apologize. It's a generic Canadian species. So this is what happens on the left if you model the distribution of that species based on the GBIF points. That's the map you get, so a map of habitat suitability. But we know that the distribution of the species, so this is just the IUCN range map, is much bigger than that, of course. So we're missing a whole bunch of information. And when we do this, we're vastly underestimating the diversity of the boreal forest. It's basically like there's no species there when we know it's not the most species-rich area of the world, but we know there's greater than what we predict there to be. And so despite me wanting to get away from SDMs a little bit, I had to go back to SDMs and think a lot about bias correction because yeah, we can't use the models as they are. It's just not possible. And so we started by taking different approaches to bias correction. Where I just started with the standard approaches that I've used many times on different continents, which usually seem to work more or less. So this is an example of some of those. So this is one model. It's a boosted regression tree, but it doesn't matter which it is. This is no correction at all, and this map is showing species richness. So it's just a sum of all the probabilities in each grid cell from the models across all species. So in the left, it's no correction. In the middle, it's sample effort correction, and this is just a really simple way of adding sample effort into the model and then making predictions, assuming that the sample effort is greater than it is. So we basically set the sample effort to maximum, and we basically get an entirely different version of biodiversity in Canada. And then the third one is the more classic approach where you have a target background sampling scheme where you're essentially weighting presences and absences based on sample effort. So you get completely different maps, and so we wanted to see which of those, well, first of all, how different they were. And the mean range size, so this is the size of a species if you would like to sum across its probabilities. The mean range size increases from the no correction to the sample effort correction. So all this is an extra variable in the model for sample effort, and it totally changes the prediction. So in this case, the mean range size increases by 400% on average for a species. So that's a bit of a problem, and this is the bias you can also see, which is really clear that the bias perfectly fits its environmental gradient. So we're extrapolating into the unknown environmental space. And then this is what happens if you add climate change to these models. So if you take the model with no correction and you project it forward, this is a mid-range scenario. I think it's RCP6. But again, it doesn't really matter. You get climate change on average increases species distributions by 35%. If you do the same for the one with sample correction, you actually get a decrease. So on average, species distributions are declining. And if you do it for the third one, you get, again, an increase of 20%. So we don't actually know if species, even our projections say if species should be increasing or decreasing. So again, a bit of a problem. These are kind of two extreme examples, but in a lot of cases, I've done these analyses for a lot of places and they usually produce relatively similar results. So it's actually the bias is more, the difference in bias is more extreme than the climate change projections. So jumping to Europe, this is the only Europe part, because one of the things that we've been working on in Europe is we're going to apply to Canada. We've started to apply it now, but it's this idea of bias correction when you have some survey data. So you have presence only data such as GBIF, you also have some survey data, and it's a really nice way to put everything together. So I won't go through it in detail. If anyone is interested in this, we can discuss it later, but this is some modeling from one of my students, Abby, and she has fit models for about 12,000 plant species for Europe. So the aim was to model all of them, and of course we can't do all of them, but she has about 12,000 species, and she has all of the GBIF data that she's filtered and everything, and then all of the EVA, so the European Vegetation Atlas. So the EVA survey data, and so she's taken where we do have EVA data, we're sort of using that as kind of a gold standard to say, do we have sort of true zeros, right? Because it's survey data compared to GBIF, where we don't know if the zeros are true zeros or not. Okay, so what's that? Oh, I wanted the other one. Before it was so much, anyway, it doesn't matter. Okay. So on this side, it's really lag, I'm just not gonna use that. So on the left side is 4 species that have only GBIF data, they have nothing in EVA, and we make a model of that, and then we compare it to the EVA models, and we basically make a model of bias for each one, and then we kind of back correct the GBIF models for this bias, so we can essentially adjust the probabilities based on this bias correction. And the bias correction happens both at a location level, so there are biases associated with location, like distance to road or general sampling effort that differs in different regions. And there are species level bias, and for this we use traits like how conspicuous are the flowers, or what is the plant height, things like that. So we have both of these bias correction kernels, and then we can adjust these probabilities. And the other part of this model is that because we have EVA as in plots, and we want to predict to a 1K grid cell, we also have a bunch of scaling factors in this that say, how do we scale up from that, from those plot sizes up to the 1K grid cell? So that's another sort of part of this model. And so what happens, which does apply to Canada as well, so this is the true EVA data. So this is the EVA data. If you look at summed surveyed presences per location, again in 1 kilometer grid cells, this is what it should look like according to EVA, and also intuitively, this is what normally looks like. In B, we have what happens if you use the predictions from GBIF. So it looks a bit weird, in classic, classic studies of vegetation, you don't normally see a bimodal distribution, and also the numbers are very different as well. And then these are the adjusted models. So after we've run the bias correction and it looks at least somewhat like it should, the numbers are approximately correct. So, and then I'll just show you the map. Sorry, the map is 2 different color scales. I really apologize. So this is on the left, it's models that are stacked for GBIF presence only. So these are kind of the more typical maps that you would see because most studies use this approach, where there's the max diversity there of about 1,500 plant species per 1 grid cell, which is a lot. And this is the bias corrected version of it. And you can see the patterns change a little bit, but more so the numbers. So here the max is about 600 plant species in a 1K grid cell. So they look pretty different. If you scale up, if you start to increase the resolution, then eventually you get the same thing, right? If you increase the spatial resolution to about 250 kilometers, then they match. But at a 1K scale, which I think is pretty useful for a lot of conservation to know how many species we have at a 1 kilometer rather than these larger spatial scales, you have a very, very different answer if you bias correct versus if you don't. Okay, so back to Canada. Someone just tell me when I have talked for too long, because I don't have any. Is it still 45? Okay, okay. Okay, so the other big project that we're working on, mostly we're working on bias correcting models, but also we're working on the other problem, which is we need to know something about the prey of the species. We need to know something about the food web to be able to understand if these species can actually move north or not. And so one of the other big projects that we've been working on for a few years now is making a large food web for terrestrial vertebrates. For Canada, it's very, very similar to the one that I worked on in Europe, and some of you have worked on it as well. So where, for every vertebrate species, we have what are the predator-prey interactions of those species. Usually they're binary or maybe 0, 1, and 2, where we have do two species, are they likely to interact? If so, they get a 1, and if not, they get a 0. In some cases now it's a little bit more refined. So we have the things that are more essential food web links compared to the things that just could potentially eat each other if they came into contact. So for Canada, because we already had the European metaweb and a fairly similar habitat, the easiest thing to do is make a model, right? To be able to predict the Canadian metaweb and then project that, to predict the European metaweb and be able to project that forward to Canada. So that's what we did at first. So one of my students, Dom, he started using kind of trait matching models. So trying to predict the presence of a food web link based on the traits of the species. So things like the match of body mass. So are the two body masses, allow for a predator-prey interaction, plus is there activity, time overlap, etc. So he built these models, and then he started to try to understand how far can we extrapolate with these models, because we're doing a big extrapolation. And so he started with small extrapolations, like what happens if you fit the models in the Mediterranean in Europe, and then you predict them to the boreal forest. So we did a bunch of cross validations with very, very strong spatial separation and environmental separation. And the models actually worked surprisingly well. We thought that they wouldn't necessarily, but they worked pretty well for about 60–70% of species, and the other species, they don't work very well, so we need additional information. But overall, you get AUC with these models of 0.95 fairly regularly. So they almost always predict the ones really well, but then they kind of over predict them as well. So if there is a predator-prey interaction happening, we can usually identify it almost always, but we also identify a bunch more that don't actually occur. Okay, so we took those models and then we projected them to Canada, and we accumulated all of the predator-prey information that we could, which was not that much actually. So there's just not much documented predator-prey interaction data, but we got all of it that we could, and then we needed more. So what we did is we looked to each national park, and we're slowly filling in this database, but we have each national park, we have a species list for each national park, and then we have people sort of making these, we're not having them recreate the whole web, but we're just sending them particular interactions to say if they're realistic or not. So far what we have is just a bunch of disagreement, not completely, but So far, it's the same problem with the models, because with the models we have this where we can predict the interactions really well, but we overpredict, and it's the same when we ask experts. So it's kind of biased in the same direction. Yeah, so we have two levels right now, the Canadian MetaWeb. So we have one MetaWeb, which is, they're all predictions, they're all predicted probabilities, but we have one MetaWeb that is the predicted probabilities based on essential interactions. So these interactions that someone that knows something has told us that they're definitely essential. So it's not globy or anything like that. And then we have another version of the MetaWeb, which is the probability of potential food web links between the two species. Okay, so once we have those, it's really nice because then we can match that up to the distribution data, like we've done in Europe before, where we take the big meta web and then we say, well, what's the species composition in each grid cell or in each community or in each national park, whatever it is, it doesn't matter. And then you can extract a local web for each of those locations or each of those raster cells. Okay, so you can get, for each predator, you can get the number of prey species, for example, that's in each pixel. Again and again, it can be either the potential interactions or the more realistic essential interactions. So the first thing that we ask with this data, it's going back to this question of will species migrate north or not. The first project is just trying to understand, will prey availability actually limit the northward expansion of Canadian vertebrates, given the data constraints that we have. So the first thing we did is just we tested, well, what happened? What's the prey distribution like for each predator in its current distribution? And then what's it like in its future distribution? So where it's colonizing to, is that different? Are there going to be a similar amount of prey when it's moving north compared to where it currently is? And for this, we have two different scenarios. So one scenario is that the predators move, they follow their climate projections, but the prey don't. And the second scenario is that every single species exactly follows its climate projections up to a maximum of 500 kilometers, because 500 kilometers is actually quite a lot to move into 2070, which is when their projections are. So you can see that this is the mean prey species richness. So again, it's based on some probabilities. This is the mean prey species richness across all predators for the present distribution, for the future distribution, and the future distribution includes what the present is as well. And the colonized is only the new stuff. So it's only the cells that the predator colonizes to and not the cells that it was in previously. And so you can see in either scenario, of course, we expect the first scenario to be a drop, but it's quite a drop. So it's on average, species have about 3 prey. On average, predators have about 3 prey in grid cells, and that drops to a little more than 1 as they're moving northward. It's a little bit less than that, but still a decline even when you let prey fully move. And the reason is because prey tend to be smaller than predators, right? So they can't move as much, basically. So it takes a lot longer for the prey across all predators to move northward. So it might not seem like a lot. They still have some prey there, but this is again on average. So many species have no prey. Many of the predators have no prey as they move northward, at least under current conditions. And so also given the sheer number of species that are likely to be moving, the prey might be a realistic constraint based on these analyses. You kind of need to work that out, how realistic that is, but just understanding that they're going to have way fewer prey as they move northward. This is an example. This is for the lynx. This is just showing what its prey distribution looks like. So you can think of this as like a prey layer. So this is the summed prey for the present distribution. So it's the lynx's distribution, the probability of the lynx's distribution times the probability of all of the summed prey for the present. That's on the left. And then what happens, and this is just its colonized area. So this is the area that it's projected to colonize into, but that is not currently. And you can see they're totally different, right? There's much fewer prey, obviously, in the north. The lynx is a really bad example because they don't eat that many prey. It doesn't. The other ones are more generalist. Okay, so that was one project. The second project, we wanted to understand how vulnerable these northern ecosystems are. So a lot of the work that we're doing right now, we're trying to help assess the vulnerability of these northern species to climate change in particular, but also just trying to understand for these areas that don't have that many species, if you have 10% of species moving in, how might that change the ecosystem? And so we wanted to understand what is the vulnerability with latitude. And to do this, we took two different approaches. One, we calculated a more typical functional hypervolume based on the species traits of the species based on the species traits. And so we had a bunch of different traits, and we assessed this for vertebrates, for birds, mammals, amphibians, and reptiles, as well as plants and butterflies. So we have a functional hypervolume based on species traits that then we can plot out. And then as a second way to assess functional complexity in an ecosystem, we also had the food webs. So the food webs where we had both these potential links as well as these essential links. So this is what it looks like if you plot out functional richness, and this is functional richness that's defined as the volume of the hypervolume based on all of the traits, and this is for all vertebrates together. And so this is what the functional richness looks like as you plot it across Canada, and this is again with latitude what's happening. So we made this graph, and then we remade this graph, and then we remade this graph like 100 times. And we're really confused because this is the opposite of the pattern that you would expect going northward with latitude. So then we got the global data. We're like, what's going on? And indeed, if you look at the rest of it, it goes down and then goes back up. So it is true that the tropics have more functional space than the Arctic. But when you look at these mid-latitudes, it actually goes up quite a lot. So we were really surprised by this. We're still kind of surprised by this. This doesn't happen if you build your functional space based on a dendrogram, because the dendrogram, every time you add a species, you're adding branch length basically. But with hypervolumes, this is what happens. The butterflies and the plants look normal. So this was fine. This is reassuring that something we're doing is not completely wrong. So for the butterflies, especially because there's most of the diversity of, you don't get butterflies really far north. So that does what we would normally expect, where functional richness declines with latitude. The plants also, you can see a steady decline with latitude. So the reason we think, and I'll just show you this next graph actually, because this is kind of useful as well. So we then looked, okay, well, what's the functional contribution of each species individually to that hypervolume, right? So how unique are the species in functional trait space? And so this is across ecoregions. So it's each ecoregion is summarized, and then this graph goes across latitude from low latitude to the high latitude areas, each point being an ecoregion. And this, for each of those ecoregions, is the functional contribution of the species in that species pool. So in the left side, we have mostly low functional contribution, a little bit of high contribution, but then as you move northward, you start to get many more species proportionally that contribute a whole bunch to the functional hypervolume. And so the reason we think is that in the north, you have two things. You have the really, really large, both prey and predators. You have the large ungulates, you have the polar bear, for example, all of these things that are really, really large, and proportionally, most of the species are much larger, right? That's what we expect going northward. But also you have a bunch of really small birds that are basically adapted to flying really long distances because they just leave, right? So you have a bunch of migratory species which are pretty small. And when you put those two things together, then you actually have a really large functional space because you have these really huge mammals, and then you have a variety. It's not only small birds, but you have a small bird species. So we think that's what's driving the pattern. So then we thought, well, what happens then if you lose some of the species? Because we know quite a few of those species are already listed as being threatened, and probably a lot more are actually threatened that haven't been currently listed. So what we wanted to do is just say, well, what happens if you pull species out of these communities? Again, across so this in this example, on the top is ecoregion and on the bottom is ecozone. Again, changes with latitude. So this left side is just a random. If you just randomly pull out 10% of the species in the species pool, you get no particular pattern. The ones in the middle are the species at risk. So these are species that are already listed according to the Canadian listing process. And you can see there's starts to be a pattern emerge. So if you just pull out the species at risk, which you wouldn't necessarily expect there to be a pattern in functional diversity, but you start to get more of a loss. And this is the change in interaction diversity. So this is just the number of food web links you have in every community. So as you pull out these species at risk, you start to lose more of your functional space or lose more of your food web as you go north. And then for megafauna, this is where we just pull out the top 10% of the largest species, whatever those are, from the community. And that you, of course, get a very, very large decline. So you get more than, and it's for some of these ecoregions, you get more than a 40% loss in your food web links. So which is a very, very large, so you're losing proportionally, you're losing almost half of the links of your food web if you're pulling out only 10% of the species from the community. So the northern ones are definitely more vulnerable when you think about it from this perspective. That was the graph before was for potential food web links. This graph is for the essential food web links, and the same thing happens basically. So we know it's not just because we've had these potential webs. Even when we say only these species have links that are really these essential interactions, the same thing happens. And especially if you get north of like 60 degrees, it really, really declines. Any questions on this part before I switch? Yeah. No, because if you did, then your random would look like that too, right? This is just totally random. There are also random scenarios that are matched up to the other two scenarios to standardize for species richness. But it's still proportionally, right? Like, yeah. Yeah. But it's true, it would, you know. But yeah, you wouldn't necessarily expect, like if you're pulling out the same number of species, you still wouldn't expect necessarily to have a trend in functional loss. I don't know. At least the randomizations don't. Any other questions? I'm going to change topics. So, you guys are quiet. Yesterday was like an hour and a half at this point. Okay. Okay, the last thing I want to talk about, and again, I'm here all day, so I'm happy to talk to anyone afterwards as well. So the last thing I want to talk about, which is something that, again, is happening in Europe as well, but we have these international targets, which in the case of Canada, we're very, very far behind. You're doing a lot better here in particular. So these are the targets for protected area. So the amount of, this is terrestrial, obviously, the amount of protected area that we want to have in each country. So the 2020 target was 17%, right? And the 30 by 30 target is 30% of land conserved by 2030. This is where Canada is. I show this graph when I'm in Canada to like emphasize that they need to pick up the pace because they're behind the US. So Canada right now has about 13.5% of the terrestrial area protected, which is completely ridiculous because Canada is this developed country that actually has a lot of land they could actually protect. Anyway. So they don't know because they wanted to know, so they want, you know, recommendations on where to expand protected areas. So we've been working on this a lot, just trying to use all these species players that we have to estimate how much biodiversity is actually protected in this 13.5% of protected areas. And so we ask for, again, all the same species set, so all of the vertebrates, butterflies, and plants. So we're trying to get a number, so how much is currently protected, and then how much biodiversity could be protected if you are to expand the protected areas in a way that would benefit biodiversity. So I won't go into this too much because it's a little complicated, because we have a lot of models and we have a lot of uncertainty. And so generally speaking, what we're doing is we're saying, if there's an area for each species, if there's an area that's stable, so we expect the species is there now, we expect it to also be there in the future. It's a very conservative approach because we're basically not weighting the areas that it could colonize to. We're just saying we really want to prioritize these stable areas. And so these areas get effectively weighted about double of the other, of where the species is now or where it could colonize to in the future. And then what we do is, in the spatial prioritization, we put all of the layers in from all of the models and we connect them. So we connect for each species, we have its present distribution and its future distribution, and it's connected in the spatial prioritization. And then we just let the prioritization run and identify the areas that are the most efficient to protect, adding to what's already protected in the protected area system. Okay, so this is the results. These are the numbers. So if in the current situation, about 15% of biodiversity is protected. So what I mean by this index, it's weighted endemism. If you're familiar with that literature, it basically sums across all species. You can think of it as an average. You can think of it as the average spatial distribution of each species that's protected. It's of course an average, so some species are 100% protected in protected areas. Some species none are zero. I think one is like 1%. But on average, about 15% of the spatial distribution of a species is protected in protected areas. So given that 13.5% of land is protected, this is roughly equivalent to just a random, if the protected areas have been randomly assigned. The protected areas as it stands in Canada do not really benefit biodiversity more than they would be if they were just randomly allocated. But if you are to expand to the 2020 target, so that's increasing 3.5% of the land, then you get a huge biodiversity gain. And we always see this. We see this at the global level. We see this within countries. Almost always with that, if you're expanding a little bit, you can get a really big gain for biodiversity. So in this case, 36% on average is protected. So we're more than doubling the amount of biodiversity only by meeting our 2020 target. And then if we were to do the 2030 target, which is never going to happen anyway, but then we could get up to 60% of the distribution if this is protected. And this is where the areas are. So anything that's in dark gray is what's already protected. And you can see the problem. It's not necessarily a problem, but a lot of the big protected areas are a little bit further north because it's easier to establish them there compared to where the people live. But the biodiversity is mostly where the people live. So that's why you see all of this along the south, where you have, so this is the ones in the magenta. I did not pick these colors, by the way. The ones in the, Isaac loves this color scheme, I don't know. The ones in the magenta are the ones for the 2020 target, and the ones in the purple are the ones for the 2030 target. And so that's not surprising. Basically, if you protect Victoria and a lot of BC, you're going a lot of the way towards optimizing biodiversity protection. But there's also a bunch of really important areas in the north. This is driven by birds. There's a lot of really important breeding grounds in the north for birds and for a lot of bird species too. There are many, many species. So again, we think of the north as not very diverse, but there are a lot of migratory species that summer there. So these are the areas that you would go. Those obviously change. So this is everything together. These change a lot. If you're worried only about amphibians, you're only worried about southern Canada. pretty much all they are. Same with reptiles. And so we have these different scenarios, and we've weighted in a bunch of different ways, based on this, trying to get rid of this edge effect. We've looked at threatened species. We've looked at functional diversity, phylogenetic diversity, basically everything we can think of, but mostly the priorities are pretty stable. So mostly these areas that are important remain important. The problem is many of these areas, and the reason there's white here is that anything that's really high human footprint, we just remove from the analysis because it's really just almost impossible to protect. And so these areas, they're kind of outside of major cities, but still in the diverse areas, especially the grassland. They're really, really important. Even if they're small patches, they're really, really important to protect in terms of maximizing the biodiversity conservation. Okay, so I'll stop there. And again, I'll get any questions about any of these projects. So first, I want to thank the team for building a Canada web. So we've been working on it for a few years. There are a lot more people than this involved, but this was our main team. And then my graduate students who have been working on SDMs, even though that was not what they were promised when they came into the lab. So Abby, doing all bias correction. Isaac's been doing all the protected area scenarios. Noah is a mathematician, so he's working on his bias correction things, and Darren just does everything. Anyway, thanks so much for listening, and yeah, I'll take any questions. Do you mean just in general that species might be moving, or? Yeah. Humans might move north, yeah. Yeah. Yeah, that's a good point. Okay. I don't know the answer to that question. So this was a really big debate about how we take into account these northward moving species. And one of the reasons we took the conservative approach, which is just to say where are the stable areas, is because a lot of the people that we talk to are very against these species moving north. And so the idea that you prioritize an area that you think will have more diversity, they're just like, no, like species moving north are evil. We don't want them. We're not even considering this. So that's why we thought, the areas of stability. In terms of the people moving north, I don't know. There's like who was it that made that human niche paper? Was that you? Someone like Catherine made this human, where they modeled the human niche over like, the last 10,000 years. And the interesting thing is that the suitable habitat for humans is about at the border of Canada right now. But by 2070, it's like almost the boreal forest. So it's good for me because I hate winter. But like, yeah, it's going to become much more habitable for people, and there's plenty of area for people to move. The boreal forest is pretty swampy at the moment, so maybe not. Yeah, maybe not. But yeah, it's true. And I think the point is that things are dynamic, and especially in Canada, they're very dynamic. So establishing a protected area set is a bit of a gamble. One of the things that we're trying to do is, because they already have two big corridor projects, there's Yellowstone to Yukon, and then there's another one that goes through Quebec, which is smaller. But we are trying to use those because those are stepping stone models, right? It's just computationally a little harder to have right now with all species in, but ultimately that would be the goal. So you can have like area that the species move through the landscape. Good question. The mic's
I just Is it working now? You can hear me? Okay. I wanted to ask, so the 58% means like a 58% more of biodiversity is protected at the 30 by 30, or is that species range? I didn't understand what the 58% refers to.
So yeah, so it's a good question. It's not 58% more, it's 58% total that would be protected. And it's like this metric, like I was explaining, the spatial distribution of the species that's protected on average across all species. So again, it's very different. So what happens in these expansions is that it targets rare species, of course. And so in the case where 58% of total diversity is protected, all of the rare species are 100% protected almost always. And then the common species are still like 10, 20% protected because it's impossible to protect a large amount. But the metric is it's weighted endemism. They now call it like species protection index, but you're basically summing the proportion of the distribution of the species that's protected across all species. So it just makes this metric, which is nice because it ranges from 0 to 1. So at 100%, the spatial range of every single species would be 100% protected. At 0, none of it would be. Does that make sense? It's a little, yeah. It's a bit, it's hard because we want a number, but it's still like, well, it's an average. Mhm. Yeah. Oh, sorry.
Okay, so thank you for this really nice talk. A lot of cool things happening in your lab. I was particularly interested in the bias correction. So we know that there's a bias in species sampling based on traits. Some species that are the cutest or the biggest, they have more sampling effort. Did you consider somehow waiting for the traits of these species?
Yeah, that's exactly what we did. So there's the two versions of the bias correction. One is the locational version, and one is the trait-based version. So what it does is it measures the discrepancy between the models predicted from GBIF compared to the EVA. So it's like how wrong it is as the response as a function of trait values plus whatever else about the species. So we wanted to get at popularity, but so what we looked at with popularity, so we have like the conspicuousness of flowers and things like that. With popularity, we basically used Google Hits, which was not important, oddly. I think that maybe that's just for plants. I don't know, because it is globally, it's important. You know, you have these studies on the popularity of species, which does influence usually whether or not they're listed or not beyond what you would expect. But yeah, for some reason, when the Google Hits didn't matter. And actually, the locational bias kernel was much, much more important than the species level kernel as well. So the traits helped a little bit. I think explained deviance was maybe 10% higher or something like that, but not nearly as much as the location, where it really, really changes the predictions.
So with this map right here, how did you select which areas would be the expanded protected range? And how does that match up with what might actually be protected? I mean, you know, so would it be easier to protect other lands that maybe aren't capturing this idealistic thing? And that's maybe assuming that you selected areas based on, you know, more bang for your buck, so to speak.
So the way that these are selected is based on the optimization. So we say any pixel that is not currently protected and is not really, really high human footprint index. So we remove cities basically and really, really intensive agricultural areas. Everything else was fair game. So anything else, you know, we have the cell, we know from the species distribution models, what we expect to be there. And then we say, well, we know what's already protected. And we use the same metric where we have, how much of the percent of the distribution of each species is in the protected areas already. Take that information, run the optimization to then say, what's the next best pixel? So basically, like if you were to go to one place, where would it be to best complement what's already protected? So it's totally based on biodiversity, which means it's totally unrealistic. And it's not, fortunately, other people are doing other smart stuff. I was just responsible for the biodiversity part, so I don't have to do the realistic part, but it was just to show like what's possible. But then if you put in the next most realistic constraint is cost. So it's cost of being able to conserve this. There are also a lot of other scenarios that I don't show here, but they're like if you were to change the management status within, so they have a lot of provincial parks that could be elevated or not, and that's mostly what they're talking about because of the government level. That's the easiest to do rather than buying up a bunch of extra land. That's really, really expensive. So there are those kind of scenarios as well. But if you put cost in, and we only have so far, the US has this beautiful cost layer that's amazing and modeled all of these fancy models. Canada has nothing, and we can't even get data. It's awful. So we just have a really, really rough estimate of cost, but it does change things a lot. And actually, a lot more of the north becomes very important when you put cost in, for sure. Yeah. So it's the unrealistic biodiversity version.
Yeah, so this question is going to be a bit vague because it's going back to the second part of your talk. And there you had these different scenarios that you considered, and it was mostly based on species. Now, I realize in your system this might be hard because you use the traits to build the models, the networks. Yeah, no, the network stuff. So in the second part, you were sort of suggesting, all the plants, not the plants, you're not studying hummingbirds, but sorry, all of the prey were suddenly gone. sorry. That's just, but all your prey have suddenly been removed. What do the predators do? Or the predators can move, but the prey can't, which is potentially a reasonable assumption. But you're looking at that at a species perspective. Now, if you were to look at that from a trait perspective, did you try to do that, or do you think it would be useful? And I realize, so maybe there's a complementary species, but in terms of traits.
Oh, sorry. So we didn't do that exactly because in the models, they're based on traits mostly. Yeah, and we know that they're over predicting a little bit. So basically, like even for the essential models, they still over predict a little bit. So basically that means for like a given species pool, you're saying that two things probably interact when maybe they don't a little bit. So you're going to get some of that complementarity wrapped up in it. So if you lose one species, you still might have another one that's considered to be there, but it might be a little bit of prey switching. It's a very indirect way of doing it. But yeah, we could look at in that functional space as well. So we could say if you pull out these species, yeah, do you have equivalent species that are still there? I think for the north, it still wouldn't be because they're quite a different diverse set of species. So probably you'd still have bigger losses, but not as much. You wouldn't have 40% of the food web disappearing in that case. But yeah, we could look at that. Yeah.
I was surprised to see during your talk that we didn't see any whales and marine ecosystems or freshwater ecosystems. Is it because of the data, missing data, or the models used, or because a large number of countries are increasing the protected areas in marine ecosystems? It seems to be quite not easy to put in place, but at least to consider.
I have this question every single time I give a talk, and the reason is because I know nothing about marine systems. That's the reason. It's not the only reason. It is a little bit harder. The data, so I've had marine people come to me and want to do these similar analyses, but the data is pretty sparse. So we haven't quite figured out, and because they move a lot, right? You know, the whales, like, so it's hard to replicate kind of this exact analysis without taking these other like factors into consideration. But yeah, we should for sure. And some people are working on it. It's just a little bit harder to get these numbers because they really want, you know, numbers where they can say how much of the distribution is protected, but when everything's moving, it's a little harder. So, but yeah, it's true. Whales are important too. It was just a place to start, right? But yes, in the future, it definitely should be. I would love to see the marine. Yeah, exactly. I know, but they're in the water. It's totally different.
If I were interested in having like a globally applicable sampling, GBIF sampling correction, what would you recommend? And obviously you have your EVA analysis, but that's kind of a regional model or refinement. If people would be interested in, for example, modeling threatened species using GBIF all across the globe, what would be the best way to sample correct?
So if you're stuck with GBIF, you only have a few options. So I guess my answer to that is I think you really can't in a lot of areas with GBIF yet. In a lot of areas, like Europe, even Europe had a lot of spatial bias in GBIF. Other places have way, way more. So I think if you want to correct GBIF, just drawing in GBIF, making distribution models, I think your only thing is to either use like I did the two things where I showed either the sample intensity, which people do is usually like a neighborhood kernel of all GBIF observations, or you do something, you know, like you're weighting your presences and absences. The problem when you start doing that, so if you think about it, what you want are all the points that are in the areas that are sparsely sampled. To make that equivalent, you can either make your other data sparser, but in doing that, you lose all of your data because the problem's so severe. So that approach doesn't, and I think that's why the target background doesn't work super well in these areas. They're just too sparse. Like there's just not enough information to fit the models. But those are kind of your two options. But I think what's nice about this bias correction is that you just need a tiny bit of survey data. And we haven't formally done this analysis yet, but you can say like how much survey data do you need for different types of species to be able to correct? And it's really not much, it's quite efficient. So I hope that, and also in GBIF, they do have presence absence plots. It's not easy right now, I think, to figure out which are which. But what I would really like to see is globally for GBIF to put that information out as available so people could do that. So they could say, well, these are our plots that we believe the absences, and then these are the points that are present only. And I would really like to see that because I feel like that might be enough information to help correct. But yeah, if you can't fit a model on no points, right? So it's kind of just an inherent problem with GBIF in these areas. I mean, again, GBIF is great. It's the best we have, right, in most places.
So I want to.
And just to remind everybody about the next events, we have Journal Club tomorrow at 3:30, and the paper is actually kind of along these lines. It's about bird biogeography, and so we encourage you to join that. And then in 2 weeks, we have another seminar, and look for that online, and you'll also get a message about it. So thanks a lot for coming.